back. All right, that was really bad. I'm gonna have to edit that out. All right, you guys, we're still in the same cell cycle section of our review. I want to talk about the cell membrane. You need to know a little bit more about the cell membrane than just its function. You need to understand how the structure leads to its function, okay? So first thing, I want somebody to raise your hand and tell me, so here's your nucleus, here's your cell. We are zooming in on this part of the cell membrane. What do I see? What is it made up of? Good, now I don't know who to call on, it's too confusing. Go ahead, Tad.
Phospholipid bilayer.
It is a phospholipid bilayer, meaning there are two layers of it, okay? If I want to draw one phospholipid over here, what does it look like?
Hydrophilic phosphate with a hydrophilic tail.
Good. So it has a hydrophilic phosphate head. So this is just a phosphate group. Phosphate. It's the same kind of phosphate you find in ATP. It's the same kind of phosphate you find in DNA. It's just a phosphate group. It is hydrophilic. What does hydrophilic mean? Raise your hand.
It's like attracted to water.
It's attracted to water and water is attracted to it, okay? So hydrophilic, hydro means water, philic means loving. So it's a water-loving part of the molecule. Then I think Emily also said it has a hydrophobic or two hydrophobic tails, and that is true. Hydrophobic. What is meant by hydrophobic? Raise your hands. It repels water. Hydrophobic, it's afraid of water, right? Water fearing. What is the tail actually made up of?
Fatty acid.
Fatty acids. And like I told you the other day, all lipids repel water. It's one of the characteristics of lipids. So it does have a lipid tail. These are actually fatty acids. All right. So I told you there are two layers of phospholipids in your cell membrane. How are they going to arrange themselves based on this information?
Tails facing inwards and heads facing outwards.
Yeah, the tails are going to face toward each other and the heads are going to face out, either outside the cell or to the inside of the cell. Why? What's on the outside and inside of the cell that causes it to arrange itself like that?
Water.
It's watery outside the cell, and your cytoplasm is a watery substance. So if there's water inside the cell and water outside the cell, it's kind of like if you take oil and you put it in a bottle of water and shake it up, you have all these little oil droplets. Eventually those oil droplets are going to come together because they're trying to stay away from the water. It's the same thing here. Your fatty acid tails hate water so much that they face inward to escape the watery inside and outside, okay? So your phospholipid bilayer is arranged like this. So the inside is the hydrophobic part and the outside is the hydrophilic part. True. What else do you see in the cell membrane?
Proteins between the two layers.
Lots and lots of proteins. You have some proteins that span the whole membrane. You have some that are just on one side or the other. They have many different functions. What are some of the functions of those membrane proteins? Keep going.
To allow certain nutrients in and out of the cell.
Some of them can actually be like channels that let certain things into or out of the cell. True.
And I was going to say a specific one. I think it's aquaporous, transfers water through.
Good. You do have a type of protein. It's called an aquaporin, and that allows water into and out of the cell in large amounts. Water can slip through the membrane, but not in huge amounts. So if the cell's trying to get rid of a lot of water or take in a lot of water, it can go through these channel proteins, your aquaporins. True. Any other functions for some of these membrane proteins? I mean, the list goes on and on. There are a few of them. All right, one other thing you need to know about these. These membrane proteins very often is how cells communicate with each other. If insulin is made by the pancreas, the only cells that are going to do anything because the insulin tells them to will be the cells that have the receptor protein for insulin in their membrane. It's also why certain viruses infect certain cells and not others. We talked about this the other day as well. If you have a nasty cold and I don't have the protein that's the lock and key fit for that virus, it's never gonna enter my cells, I'm never gonna get sick. Alright, so these proteins have a lot to do with communication or recognition between cells, okay? What else do you find in the cell membrane?
Cholesterol.
You do have a little bit of cholesterol. What type of organic molecule is cholesterol? It is a type of lipid. Doesn't look at all like your fatty acids, but it is a type of lipid. You do have cholesterol in animal cell membranes, specifically. Why? What's the function of that cholesterol in the cell membrane? Here's when I hit the crickets button. Function of cholesterol in cell membrane. Alright, you're all writing it down. It actually gives the cell membrane a little bit of stability and support. So it's not, it's a little more maybe rigid than just a fluid membrane would be. It's also the reason why you don't really find cholesterol in the cell membrane of plants. They don't really need it, they have the cell wall on the outside, okay? Go ahead.
So if you were, if you had like a disease where you weren't immune to like many, like if you, would that mean that you had a lot of proteins in your cells or in the membrane or like none at all?
There are so many different types of proteins that can be in, on, and around your cell that it really depends on the type of protein. But there are some diseases associated with not having the correct membrane protein so that cell doesn't respond to signals that it's supposed to. Yeah, there are quite a few diseases like that, definitely.
What is the cell wall made out of? Because like you know there's water on the other side of the membrane, and right next to the membrane is the cell wall in plant cells. So is it made out of water?
Nope, remind me, what's the cell wall in plants made out of?
Cellulose.
Cellulose, and what type of molecule is cellulose?
Carbohydrate.
It is a carbohydrate, so it's just a whole bunch of sugars chemically stuck to each other, so water can pass through the cell wall without a problem.
So is there like water in between the membrane and the wall?
A little bit, but you want to really think about that membrane as sort of, especially if the plant has enough water inside the centralized vacuole, that membrane's really sticking up against that cell membrane, but sure, you could have a little bit in between.
Okay.
Did I answer your question?
Well, okay. So you know how there's like the structure of the thing is like the heads and the tails, and they face a certain way because there's water on either side of it.
So you're looking at this, but now you're talking about a plant cell that also has a cell wall around it.
Yeah.
It would actually be more like
would
this
it-
because it's around it. Okay, go
Would
ahead.
it not make that structure because there's a cell wall right
No,
up
it
against
still would because it is still a watery outside and inside. So I see what you're saying. You're wondering if there's water inside here to make a watery outside. And the simple answer is yes, but there's also still plenty of water out here. You don't want to think about this as ever controlling what's coming into and out of the cell. Lots of stuff can pass through the cell wall without a problem. So you definitely still want to consider it a watery outside. So yeah, it still arranges itself like that. And this can be simulated in a lab. There are scientists that can take phospholipids and put them in a beaker of water, and the phospholipids arrange themselves in a bilayer naturally because of the hydrophobic and hydrophilic parts. Okay. Now here's where my concern is for some of you guys answering open response questions. MCAS loves, and I love this too, I think it's really important, loves this type of question. Describe the structure, and part B, relate the structure to the function. So we already said the function of the cell membrane was to let certain things in and out. We talked about endocytosis, exocytosis, but how does this structure actually allow the function? You guys want to keep thinking about that in biology.
Oh, its semi-permeable structure allows it to take in the nutrients and other chemicals which it needs in order to go about the cell's functions.
True, it's semi-permeable, so certain things can be allowed in, certain things can be allowed out. What kinds of certain things? All right, what if I wanted to get a huge protein inside the cell? Is it going to be able to easily slip through the membrane? Yes or no? Why or why not?
What if you had
Yep, hold that thought, that's where we're going. Is this big, please don't forget that thought, all right? This big protein, is it slipping right through the membrane? Yes, no, why or why not?
No, because it's too large.
Simply because of size, it is way too large, it's not just slipping through. It would need to come in by endocytosis or through a specialized protein, definitely. Go ahead.
If the protein was surrounded by a vesicle, if the vesicle bonded with the cell membrane, couldn't it have just...
Of course, absolutely, absolutely. And usually the cell membrane will actually grow that vesicle around it to bring it in, but again, that's part of the structure of the cell membrane. It's able to sort of grow those arms and create a vesicle and bring that material in because the phospholipid bilayer is like a fluid, it's movable, it's flexible. All right, so that does relate to its structure, absolutely. What if I wanted to get a molecule that's like a sodium ion into the cell? It's small enough to slip through the cell membrane. Is it going to slip through the cell membrane? Yes or no, and why or why not?
No,
Why?
because it's positively charged, and so it would need something to like counteract.
She's on the right track. Her answer was no. Now I'm being recorded. Having a coughing fit. Excuse me for a second. Oh, goodness for that little sip of coffee. All right. The answer was no, that sodium ion is not going to easily slip through the cell membrane even though it's really tiny. And the reason is this. By hydrophobic, remember, water is a polar molecule. It has a slightly positive end and a slightly negative end. That means it loves things that are charged. So the sodium ion can get past the heads because they're also charged, but they're not going to get past this lipid interior that's hydrophobic. It's not going to be able to slip through there because it's so charged. All right, so usually ions need to come in either through protein or through active transport. Okay. Questions about that? All right, so we've been throwing around active transport versus passive. Pure vocab here. What's the difference? Raise your hands.
Passive, like if you just slip through, like diffuse through without any energy required, whereas active would require ATP.
Good. So no matter why, active requires ATP for something to get into the cell or out of the cell. Passive does not. So is diffusion active or passive, and why? Raise your hands.
It's passive because it can just go through the cell wall without ATP.
True, so it's passive because there's no ATP needed. Related to concentration.
If one side of the cell membrane has a higher concentration of an ion, right? And it wants to go to the other side that has less concentration to even out the balance, it'll just go, so it's an energy go against the force.
Yeah, you really want to think about concentration when it's, you definitely, first thing you want to know, active transport requires ATP, period. Passive transport does not, period. But you also have to know about the concentrations and how that's related. Here's what I mean. If outside the cell you have a whole bunch of one molecule, and that molecule can easily slip through the cell membrane, and maybe you have another molecule where you have a lot of it inside the cell, it can also slip right through the membrane. If I want to get this pink star molecule out of the cell, is that going to be active or passive, and why? John.
I want to say that's active
Why?
because it's pushing it out of the cell.
You're pumping it against its concentration gradient, all right? So whether it can slip through the phospholipid bilayer or whatever almost doesn't matter here. The fact that you're trying to go against the concentration means you have to add energy, you have to add ATP. It's no different way back when I used the analogy of 100 people in this room and one person in that room. That one person is going to have to exert energy in order to push his way or her way through this room. You have to exert that energy if you're going against your concentration. Go ahead.
Wouldn't this be a hypertonic situation with the
Yeah.
water leaving the cell?
That's a good question. We're gonna hold off on that tonicity stuff, because it is important, but I don't see a lot of questions purely on tonicity. The questions you're gonna see on MCAS has to do more with the logical thought, process behind concentration gradients, okay? So let's stick with our examples, one more. If I wanna move the circle molecule out of the cell, and it can pass through the membrane, no problem, is that gonna be active or passive, and why?
It's passive because there's a smaller concentration of it outside of the cell. So
Yeah.
it moves to the lesser concentration.
It's a natural tendency of materials to move to where it's more concentrated to less concentrated, so this would be passive. In my example of the classrooms, 50 people in here, none in there, it's gonna be pretty easy for us to just slip into the other room where it's not as concentrated.
Just a small question, since this is like mostly in water, like is it osmosis or can you still say it's diffusion?
That was my next question. What is osmosis? Go.
Diffusion of water?
Yeah, if this molecule that's just diffusing, diffusion is just that natural tendency to go from a high concentration to a low concentration. If that molecule happens to be water, it has a special name, and that special name is osmosis. But osmosis is just diffusion of water. Water is such a unique, important molecule, though, that it tends to have its own names for everything. All right, so osmosis, if this is water, that would be considered osmosis. If it's anything but water, it's just plain old diffusion.
So it's osmosis is diffusion and diffusion is part passive transport, just like osmosis is passive.
Good, osmosis is always passive transport, yes, yes. And the types of questions I've seen related to this, I see a lot of questions on osmosis, but I do see questions on the sodium-potassium pump, but you want to make sure you're really paying attention to the concentration gradients. There are some proteins that can let sodium just diffuse in or diffuse out, but if it's getting pumped against this concentration gradient, like the star here, then it would require energy, it would require ATP. All right? Any other questions about this? I only have 2 other questions on this list about transport across the membrane, and then we are going to whip through the cell cycle really fast. All right? What is the difference between endo and exocytosis?
Endocytosis is bringing materials into the cell, and exocytosis is like exerting the cell.
True, but is it doing it like one molecule at a time like this?
No, it takes them in like all at one time.
Yeah. So if the cell is taking materials into the cell in a vesicle, that's endocytosis. If it's releasing contents out of the cell, those contents travel through the cell in a vesicle, then that vesicle fuses with the membrane and it releases its contents. That would be exocytosis. You should know those 2 terms as well. All right. Other questions or comments? Who is sick of bio review? Do not raise your hand. All right, don't even smile. I can see you, you're like laughing. All right, fair enough. I'd be feeling the same way if I were you, but it's okay. I'll torture you a little more. All right, switch gears. Am I still okay there?
You're good.
Is it exploding because of my nonsense?
No.
Is it like no teacher should ever act like this? This big red warning. All right, cell cycle. I didn't even ask a question yet, go.
I was gonna say you should edit that.
Yeah, I might, however, I have a feeling that tonight I'll be lucky just to get the stuff uploaded, so I don't know if I'll be doing any editing, but maybe. And I would say Adam would edit it for me, but I think I'm not gonna let Adam see it because he will laugh at me forever, and now I'm recording myself talking about Adam. Okay, we're definitely gonna have to edit that. Cell cycle, here we go. What is the cell cycle? Raise your hand, just pure, easy vocab definition. Thank you. It is the life of a cell. Easy to think about a cell cycle, like we talked about nutrient cycles, cell cycle. You always want to think about your cycle as being a cyclic pattern, okay? Cell cycle, you can break it into 2 major parts. What are those major parts?
So basically the first chunk is interphase, and then the second mitosis, which at the end is cytokinesis.
Okay, so you have interphase, and you have the mitotic phase. Those are the 2 main phases of the cell cycle, but each of those phases can be broken down further. What are the three main phases of interphase?
G1, S, and G2.
G1, S, and G2. I don't want to spend too much time on these. We do need to talk about S though. Before we talk about S, what is happening in G1?
Isn't it just preparing for like the cell cycle?
It is preparing, it is getting, I like to think of G1, it doesn't stand for getting ready for, but think of it that way. G1 is getting ready for what? It's getting ready for synthesis, for DNA synthesis. So this is when the cell is making, it's getting ready for, it's making everything it needs for DNA synthesis, which is also known as DNA replication. So for example, DNA polymerase made in G1. Everything the cell is going to need to do S phase is made in G1. Once everything's made, S phase happens. What is S phase? Raise your hand.
The actual DNA synthesis.
DNA synthesis, DNA replication. And here's a question they love to ask in many different ways, shapes, and forms on MCAS. And a lot of you way back when lost points on an open response for this as well, okay? First of all, how many strands are there in a DNA molecule? Raise your hand. Okay, I should see lots of hands up. How many strands of polynucleotides in a DNA molecule?
2.
2. You have 2 strands. Now they are twisted, like a ladder that's twisted, but I'm going to draw them as a flat ladder here, okay? If this is part of your DNA, before S phase, T, C, G, G, A, T. What do these letters stand for? What is that? Good.
No, the nitrogen spaces.
Yeah, don't forget, phosphate, sugar, these are your nitrogenous bases, okay? And they are hydrogen bonded to each other in your double-stranded molecule. All right, so really quickly, somebody tell me the sequence of the second strand.
T, A, G, C, C, T, A.
Okay, that's your DNA molecule at the end of G1. When it enters S phase, each strand becomes a template. MCAS loves to ask that. You need to know that when you end with 2 strands at the end of S phase, each new molecule, you end with 2 molecules at the end of S phase, each new molecule is half old strand and half new strand. You have to know that. So in S phase, this gets split in half, maybe a little bit better than I split it. DNA copies itself using each strand as a template. So now this is completely separated. You have one strand over here, you have your other strand here. This is one of the most amazing things about DNA, the fact that each strand can be a template and you get 2 identical molecules, okay? So, splits in half, this is a template, that's a template. What's the enzyme that actually builds the new DNA? Polymerase comes along and it starts building one at a time. T, another one, A, adds another nucleotide, G, it adds another nucleotide, it adds another nucleotide, it adds another nucleotide, it adds another nucleotide. Now these are hydrogen bonded. This is the exact same DNA molecule as what you started with. One old strand, one new strand. Similarly, the other strand becomes a template. Polymerase comes along and builds the new strand, one nucleotide at a time. So what you end with are 2 molecules that are identical. Each one is half old, half new. Do not forget that for MCAS. And if you're asked about DNA replication, include that in your answer, okay? That is what happens in S phase. Every single bit of your DNA is copied. So then what happens in G2?
It gets ready for the actual division.
It's getting ready for mitosis, all right? G1, G2, it's all about getting prepared for the next step. So this is making everything you need for the mitotic phase.
Well, I was gonna say that some organelles are replicated if you.
Yeah, yes, you definitely have like the enzymes needed for mitosis here being synthesized, but this is where you would have other organelles being made as well, because I mean, organelles can be made throughout a cell's life, but you definitely want to start off your cell with at least a couple of each of the organelles. Good point. Kyle, do you have something to add?
No.
All right. Mitotic phase is technically mitosis plus cytokinesis. Somebody raise your hand and remind me of the stages of mitosis. Just list them.
Prophase, metaphase, anaphase, and telophase.
Prophase, metaphase, anaphase, telophase. If you have a hard time remembering that, I always like my please make another taco. That doesn't make me really hungry for lunch. Prophase, metaphase, anaphase, telophase. You need to know what's going on in each of these phases. Somebody tell me what's happening in prophase. So let me say, at this point, at the end of G2, you have a nucleus inside a cell, and every single chromosome is completely copied, but it's still chromatin, all right? So what is the first thing that happens in prophase? Somebody raise your hand. Good, I haven't lost you all yet. Go ahead. The chromatin starts to condense and you start to actually see your chromosomes. And at this point, your chromosomes show sister chromatids, copies of each other.
You begin to see the nuclear envelope break down.
Yeah, you also start to see your nuclear envelope is breaking down. Anything else?
Sometimes notice that the cell is enlarged in size.
True, true. You really want to worry about just the spindle apparatus, that part of the cytoskeleton that helps pull these chromosomes, these sister chromatids apart. Your spindle apparatus starts to form here as well. Okay? Alright, so now by the time you get to metaphase, there's no more nucleus. What's happening in metaphase? Raise your hand.
The chromosomes line up in the middle of the cell.
All your chromosomes line up in the middle of your cell. So you'll have, I'm going to draw the nucleus even though it's not up there anymore, just to sort of make it easier for you to see the middle of the cell. Alright, so you have your chromosomes all lined up in the middle. So what happens in anaphase? Raise your hand.
The chromosomes start to pull apart.
The sister
Sister
chromatids, the copies, start to get pulled apart. Alright, so your spindle at this point is attached to each center of the chromosome, and in anaphase it starts to pull those apart. So usually you see it and it looks sort of like this because the spindle is pulling it from the middle. Alright, so you're actually separating your copies, you're separating your sister chromatids. Each is going to the opposite end of the cell, the opposite pole of the cell. So then what's happening in telophase? Raise your hand. If I wait long enough, everybody will eventually raise their hands. Go ahead.
The nuclear envelope starts to like reform.
Telophase is kind of the opposite of prophase. So you are still one cell, but they're definitely, the chromatids have been pulled to one side or the other. You start to reform your nuclear envelope around each set of DNA. And
The chromosomes decondense.
they start to decondense. So instead of having this chromosome shape, you start to see that they're decondensing back into chromatin. So then what's left to happen in cytokinesis?
The cell actually splits.
The cell splits, the nuclear envelope is completely formed, the chromatin is completely, the chromosomes are completely decondensed. So what you end with in cytokinesis means the splitting of like the cytoplasm. So what you end with in cytokinesis is exactly what you started with, but you don't have double the amount of DNA anymore. I want to put some numbers on this. Don't get confused, we're talking mitosis here, alright? D block will get the lesson on meiosis, and you guys will check it out on YouTube, hopefully. If this is a human cell, how many chromosomes are there in G1? Raise your hand. Don't be shy, I won't yell at you if you, well, I probably will, but in a nice way.
46.
46. At the end of S phase, how many chromosomes are there?
92.
Why?
Because each chromosome is then replicated.
Exactly, you've copied every single chromosome, you have twice the amount. Prophase, how many do you have? Raise your hands.
92.
You still have your 92. Metaphase, you still have your 92. Anaphase. It's a tricky one because you're still one cell, but you basically have 46 at the top, 46 at the bottom. They split, and you end up with, is this sexual or asexual reproduction? Raise your hand. Good, 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 good.
Sexual?
Say it again.
Sexually?
Asexual, asexual. Sexual, you'd be getting DNA from mom, DNA from dad. That's when you'd have to go through meiosis first, so be careful, all right? I know you know this, but those are the kinds of questions on MCAS you have to make sure you get right. Those are sort of like the free gimme points, all right? This is asexual reproduction because there's only one parent cell involved, and the daughter cells, or the son cells if you prefer, are exactly the same genetically as the cell you started with. Don't be wrapping your stuff up. I have 5 more minutes with you guys, please. It's the same exact DNA, same amount of DNA as what you start with. Did you look at the clock? What? Look at the clock. All right, questions or comments about this? Honestly, guys, that's all I have. I think if I try to do any more with you, your head will explode. So you're here for 5 more minutes. I'm going to ask you to just maybe chill out, look through your stuff, and get your heads together, and then you can go to lunch. Turn that off, yeah?